Hey everyone, today I'm at the ANPC with Ruey-Leng and we're talking about Program 3 at Future Food Systems. So what's been happening at the ANPC?
Well, there's a lot been happening at the ANPC. Some of the projects are related to the Future Food Systems CRC project. In particular, we use the analytical instrument that we have at the ANPC and develop a number of different methods to profile or fingerprint the food that we eat. What I mean by fingerprint is that we are doing it a little bit more than what traditional food analysis would do, which means that we look beyond what is the carbohydrate amount or the protein or the fat amount in there. But instead, we wanted to look at what are the different chemical compounds in the food so that we can understand what's the potential impact once we consume those food.
So you've been basically creating a new way to profile food. What do you think the outcomes of those will be to, say, food industry food labeling?
Well, there's a number of different things. First of all, by knowing what are those different active ingredient or bioactive component, then the food industry can then utilize those informations to tell the consumer this food is high in a specific bioactive component. For example, polyphenol or inulin, some of these compounds that has been linked with potential health impact. And as at the other angle of what we wanted to look at, other than just looking at the biochemical compound of the food, is what happens when we do consume those food. Does it make us healthier or does it prevent us from developing a disease?
So the food industry has been using inulin for many years as an ingredient, particularly in cereals and into drink and beverage products. But you're going back to the produce. What's some of the produce that you've been analysing?
As part of the CRC program, we're currently working with a farmer down in Denmark that their family has been growing these inulin-based protein plants such as elephant garlic, Jerusalem artichoke, globe artichoke, as well as yacon, for example, to look at these different plant products. The purpose for our analysis is to understand this inulin compound. Although when we talk about inulin, to us, we probably just think of about one molecule, but actually it's a very complex molecule. It's a large molecule. So what we noticed based on the method that we have developed was that the inulin compound in the yacon is very, very different to the elephant garlic. The sizes of the inulin compound in elephant garlic is much longer, and when we cook them, the impact of what happened to this inulin compound is also being affected differently compared to different products. We actually also have a small pilot study to see what happens when we ingest those and how quickly they go down to our gut and it's being digested by our gut. So by understanding and profile or fingerprint this compound more in depth, we now can understand how is that going to help us to impact our health in a more molecular level, really.